Hi, it's great to be here today to show you some data and to discuss how we use human stem cell models of neurological disease, specifically to advance drug discovery projects. Now, I'm going to talk to you about some of the work that we do in our labs at Talisman. Um, the vast majority of our work is based on iPS-derived, fully human CNS models that we generate ourselves. So we work with a range of different iPSC lines from different genetic backgrounds, and I'll come on to that in more detail. And we differentiate the iPSCs down either a neuronal lineage to generate astrocytes and different types of neurons, or towards microglia. We can then work in these systems in a very modular way. So working in 2 dimensions or 3 dimensions, um, culturing astrocytes, neurons, and microglia either as monocultures or as multicultures that may include 2 or more of these different cell types, really depending on the questions that we're trying to answer. I'll hopefully today convince you that these are really informative disease models and show you some of the different ways that we use them at different stages of the drug discovery process. These models are really scalable. Most of the data I'm going to show you is from 96 well formats, but we do go up to T75 if necessary to generate large quantities of cells. Okay, before I start on the data slides, I wanted to just give an overview of the different applications of stem cell models in neurodegenerative disease research specifically. So we do a lot of high content imaging based endpoints, and you'll see that throughout this presentation, there's a lot of nice images. These model systems are really amenable to genome engineering. So we've run phenotypic screens, both genetic, so CRISPR screens, and small molecule screens in typically 96 well format with imaging-based endpoints. A number of these have used disease-related phenotypes that are present in the genetic backgrounds associated with disease. We can use these either to try and identify modulators that may reverse the phenotypes, or to study disease biology in affected cell types. So particularly looking at trying to understand the mechanisms underlying the generation of those phenotypes. We've often used the systems for target and pathway validation, so right at the early stage of drug discovery process, to really make sure that the target that we're focused on is the best that we can. In addition to that, I'll show you some examples of where we've used these complex systems for compound profiling at the kind of late lead optimization or candidate selection stage. Again, before I move on to data, I just wanted to stop and highlight the importance of quality control. Now, QC is really important in many different aspects of scientific research, but particularly with iPS-derived systems that are extremely complex and can take many months to generate cells. It's really important to make sure that we understand what we're looking at and that we have really reproducible protocols. So we work with proprietary methods that are based on published protocols, and we've got some references on these slides to generate cortical neuron astrocyte culture systems. This in vitro differentiation protocol mimics the in vivo human neurogenesis. So we obtain reasonably mature neurons from around day 60 in vitro, and we typically study their biology and behavior from around day 60 to day 100 in vitro. So these are quite extended periods in culture. The differentiation process, while we have a very nailed down protocol, it is complex and things can go wrong. So it's really important for us to check at key points to make sure that we're on the right track, that we know that we have generated for example, cortical neurons that we're looking for. Some examples of the points at which we employ QC processes are at the beginning with the iPS lines, so really to confirm that if we have a disease-associated mutation in the line, that it is genuinely there. Also to ensure that carrier type is as we expect and that we haven't either acquired extra copies of chromosomes or kicked any chromosomes out. An important part of our in-house QC is the transcriptomic QC of progenitors. So on the top right-hand side of the slide, you can see an image, a heat map from a nanostring gene panel. And this is published, but we use this to really sense check our progenitors at around about day 30 to check that they're expressing the genes that we would expect of cortical progenitors. We also run immunofluorescence staining to make sure that we see expression of beta-3 tubulin, MAP2, for example, as shown on the bottom right-hand side of the slide, and look at the cultures really regularly. So we've got a really experienced team who know what neurons are meant to look like, or microglia or astrocytes. And it's really important to just keep an eye on whether the cells are visually appearing as we'd expect. Accurate and complete record keeping is really important. And a really key point is that in addition to running all of these QC processes, that we respond to any data that suggests that the inductions are not meeting our criteria and that they are discarded and not used for following experiments. So all of the data that I'm going to show you obviously is from systems that has passed QC. Another important part of our work has been to really characterize the systems that we work with upfront so that we understand what we're working with, we understand the time points that are appropriate for the different studies. So one part of that for the cortical neurons, for example, is looking at neurite outgrowth. We have some images on this slide showing neurons in phase contrast and using a lentiviral reporter. These data were generated on the IncuSite and show kind of static images, but the algorithms that are within the IncuSite allow us to monitor neurite outgrowth over time. Another important aspect of the characterization from the neuronal perspective is confirming that the cells are neuronally spontaneously active. We've done that using electrophysiology and MEA recordings, but on this slide, if the video is going to work for me, you'll see a calcium reporter. So every time there's a flash, this is the calcium reporter lighting up because calcium has been increased in the cells. What you can hopefully see is that we have quite a regular burst activity in these neurons, and that the culture is lighting up together. So there's synaptic connectivity within this system. The trace, the graph on the top of the slide, shows regions of interest with fluorescence plotted. So it's possible to track different regions of interest within the culture, monitor the fluorescence, and you can see that the burst firing can be shown to be synchronized. We've also run a lot of characterization to demonstrate that this is NMDA-mediated synaptic connections. We've worked with things like tetrodotoxin to show that that will obliterate the activity as well. So we have human iPS-derived neurons that we're happy express proteins that we would expect. They visually look as we'd expect. They're spontaneously active. So how do we use them? Now, human stem cell models are really powerful systems in their flexibility and the fact they're human. So for drug discovery, historically, rodent cultures were really the cornerstone of a lot of the in vitro modeling. But the ability to work on human systems obviously brings us that increased translational value, particularly where we have molecules that don't necessarily have great species cross-reactivity. Now, in addition to the advantages of working on human systems, one thing that's really powerful about the iPS-derived systems is that we can work with neurons, microglia, astrocytes from patient backgrounds. So we've particularly worked with autosomal dominant mutations that are causally associated with monogenic Alzheimer's disease and other different forms of dementia and neurological disease. Now, a couple of examples of these will be shown in the following slides, but particularly the APP and PSEN mutations, which are linked to monogenic Alzheimer's disease. Now, we've shown, and a lot of this data is published, that these neurons are actually able to recapitulate aspects of early disease pathology in vitro. Now, examples of this, some of which I'm going to show you data on, are altered beta amyloid 40 to 42 ratios, alterations in tau biology, extracellular synaptotoxic forms of A beta that are produced and released into the cell culture supernatant, and also altered endosomal and lysosomal functioning. Now, the really important point about this cellular dysfunction is that it's present in the systems that we're working on. It's directly linked to the mutations, the disease-causing mutations that are present in these neurons. But this cellular dysfunction is also detectable in patients and can be evidenced in post-mortem tissues, for example. So these systems provide a really nice way to study those early events in the disease pathogenesis, and an opportunity to try and identify molecules that are maybe able to reverse some of this dysfunction. So I'll show you a couple of examples of disease phenotypes. The first one is the altered beta amyloid ratio in some of the APP and PSEN mutation neurons. Now, many of you will know that amyloid beta plaques are really a hallmark of Alzheimer's disease pathology and have been targeted themselves with antibodies, which have now actually been approved by the FDA. The reason people are interested in targeting A beta is that the amyloid cascade hypothesis has been a real cornerstone of Alzheimer's disease research, and proposes that the accumulation of beta amyloid is really fundamental to disease progression, and that particularly the longer forms, so A beta 42, are the most toxic, and therefore the ones that you would like to clear, for example, with therapeutic antibodies. In the APP and PSEN mutation neurons, we see that the beta amyloid ratios are actually skewed towards the longer forms. And the bottom right-hand side graphs show the ratios of A beta 40 to 42 and 38 to 42, which are reduced particularly in the PSEN and APP V717I mutation neurons. And that's driven by an increase in the longer forms of disease. So this is a really nice system where A beta levels can easily be quantified in cell culture using commercially available ELISAs. And this system can be used to identify modulators of APP processing that could ultimately be therapeutically useful. Okay, the second phenotype that I wanted to introduce you to and discuss with you is the altered endosomal and lysosomal functioning. Now, this area of biology is actually shows dysfunction across a whole range of different neurodegenerative diseases, including Alzheimer's, but also Parkinson's, Huntington's, and ALS. To back up a bit, the endosomal and lysosomal systems are really the cellular processes and systems that regulate the sorting and recycling of cellular proteins. And there's a lot of evidence, particularly from GWAS studies, that implicate dysfunction in these systems as being a core risk factor in Alzheimer's disease, as well as other neurological diseases. Going back to my point that what we're really looking to do is model early stages of disease, and so model cellular dysfunction that can be detected in later stage patients. That's true of the endosomal and lysosomal dysfunction. So on the right-hand side, you can see electron micrographs, which show abnormal accumulation of lysosomal dense bodies and autophagic vacuoles in dystrophic neurites from an Alzheimer's disease patient. Now, another way of looking at this is, again, from patient neurons, is to stain for lysosomal and endosomal proteins and enzymes. And in this example, we have cathepsin B, which is a lysosomal enzyme, stained in control neurons and Alzheimer's disease neurons. And hopefully what you can see is a kind of increase in the size of the puncta that are detected in the Alzheimer's disease neurons. And this is indicative of enlarged lysosomes, particularly in the cell body. Now, this is something that we also see in the APP and PSEN1 mutation neurons. So this enlargement of both the endosomes and the lysosomes. In this particular slide, you can see LAMP1 shown in red, with images from, these are cell bodies, so the blue is DAPI, showing the cell nucleus. The top three images show control neurons, and then a range of different APP and PSEN mutations in the second and third rows. Again, what you can hopefully see is a similar story to the previous slide, where the LAMP1 staining is really forming these large accumulations of lysosomes. Now, if we take these images on a high-content imaging platform such as the Opera Phoenix, which we use regularly, we can actually quantify the area and the number of these LAMP1 positive puncta and compare either between genotypes or plus and minus various different treatments. A lot of this data is published and the references are on the slide. But what we see is the increase in LAMP1 puncta, so an increase in lysosome size in the Alzheimer's disease neurons that really parallels the observations in post-mortem brain. And we've been able to do some mechanistic work to demonstrate that it's actually lysosomal degradation that is the kind of root of the problem in these cells. So on to how we use this to support drug discovery projects. I mentioned that we can use these imaging-based endpoints and in combination of high-content imaging systems and automated image analysis algorithms to monitor the size and area of different vesicles and puncta within the cells. So the RAB5 and LAMP1 immunofluorescence metrics allow us to look at abnormalities in endosomes and lysosome size and area, and to quantify this. We also have a number of different molecules which we know will either improve or worsen the phenotypes that we see. So the top graph, you can see the effect of baflamycin, which is a VATPase inhibitor that effectively prevents lysosomal maturation and degradation. So it produces an accentuation of the phenotype. So it causes lysosomes to accumulate around the cell body. And we pick this up really nicely by looking at the LAMP1 positive spot size. We can also look at P62 and LC3 to quantify autophagosome size and area. And we have a system that allows us to look at TFEB translocation to identify compounds that impact lysosomal biogenesis and autophagy. And in this, we're looking at the ratio of intranuclear to extranuclear TFEB. And you can see on the bottom graph that chloroquine will shift that ratio towards nuclear translocation. So there are a number of different ways that we can use these imaging-based endpoints to identify molecules that are having particular effects in these different neurons, with a view to trying to reverse the endolysosomal dysfunction that's present in the APP and PSEN mutation neurons. One really nice thing about the imaging endpoints is that we can often multiplex them to maximize data output. Okay, another thing that we're able to do via live imaging is to look at lysosome function. So if we use commercially available reporters, such as the pepstatin reporter, pepstatin is conjugated to a fluorophore and will bind to active cathepsin. So it enables us to detect and quantify the amount of active cathepsin that's present in the neurons by quantifying the fluorescent puncta. So you can see in top left, the vehicle response, we have active cathepsin present. Baflamycin deacidifies the lysosomes, so effectively means there's a huge reduction in active enzyme. So that's a really nice negative control. Compound X is an example of a compound that we identified that has increased the amount of active enzyme within the lysosomes. So this assay can be applied for target validation, for candidate selection, and to generate dose responses for novel chemical series. Okay, so I just want to spend a little bit of time talking about microglia as the immune cell of the central nervous system. Now, microglial dysfunction is a really important factor in a wide range of neurological diseases, and there's large species differences in the behaviour of rodent and human microglia. So these human systems are really important. Similar for neurons, the human microglia are not readily available, so iPSC-derived systems have been a really important advance to help us really understand the role of microglia in CNS biology and pathophysiology. In the same way as the neurons, we've characterized the cells that we've generated, and so a couple of bits of data for you here show that microglia become fully differentiated. They express the proteins you'd expect them to. So in this example, CD68, IBA1, they become quite ramified. They produce cytokines in response to LPS and LPS interferon combination. We've particularly focused on TNF alpha and IL-6, but we've run MSD panels and shown a range of different cytokines produced. The cells are also phagocytically active. So the graph on the right shows phagocytosis of Frodo-labeled apoptotic cells, and this can be inhibited by a combination of cytokilasin D and dinosaur. So we're able to demonstrate that the microglia are behaving in ways that you would expect. We can quantify that behavior, we can modulate that behavior. We've also done a lot of work on microglia neuron co-culture models. So I think all of the data that I'm going to show you on co-cultures, we've also produced in monocultures. But the microglia neuron co-culture systems provide a really interesting system to understand the role of microglia in neurological disease and understand the interplay between neurons and microglia. So there's a strong link between neuroinflammation and neurological disease, and there's a lot of interest in trying to generate new modulators of neuroinflammatory processes. It's quite a complicated picture though. So microglia activation status isn't fully understood at different stages of disease. There are potentially both protective and detrimental roles that have been described. So I think there's been a real need for human in vitro systems that are quite flexible to allow us to really unpick what's going on and how microglia could be modulated therapeutically. So we work with microglia neuron co-cultures in 96-well plates, and these cultures have both 2D and 3D areas present, but they are in a 96-well plate format. So just to explain what I mean, this video shows a section of neuron microglia co-culture where the microglia are really embedded in the 3D structure of the kind of dense neuronal network. If I repeat the video, but without the blue dappy nucleus stain. Oops. Stop that one. There we go. Then you can hopefully see that we have really nice tiling of the microglia. They're very much integrated into this dense network of neurons, but have this really nice kind of more ramified morphology that we can image using the Opera Phoenix. Now, a couple of other examples, again, of microglia neuron co-culture systems. In these videos, we have microglia that are expressing GFP. You can't see the neurons, but hopefully what you can see in the quiescent microglia video is that the microglia have spread themselves out really nicely. The cell bodies are pretty static. I'll go again. But the processes are highly ramified and very motile. So they're dynamically surveying the surrounding area. Now, this is exactly what we would expect microglia to be doing in vivo. We also wanted to demonstrate that the microglia will respond to damage. So if during their surveillance behavior they detect that something is amiss, for example, the red spot on this right-hand video shows a point of laser damage. You can see that they really respond rapidly and chemotax to the point of injury. So we've been able to show that in the co-culture systems, microglia behave and exhibit both homeostatic and damage response behaviors in exactly the way that we would hope. Okay, so in terms of how we have used these systems to support drug discovery projects, I just have a couple of examples. We've obviously characterized the system, shown, as you can see on the left-hand side, that we can co-stain for neurons and microglia. So you have the neurons in green, microglia in red. Again, you can see the microglia quite nicely tiled out. As a kind of early study, we selected 10 compounds, commercially available compounds, from the literature that had been described as being either anti-inflammatory or neuroprotective, to see whether or not we could demonstrate effects just on a fairly simple LPS interferon gamma induced TNF-alpha release. We identified CP863187, which is a P38 inhibitor, and dexamethasone as being particularly good reproducible inhibitors of TNF-alpha release. And we went on to generate IC50s from concentration effect curves for both of these compounds, which are very much in line with the literature data. So this system is robust enough to produce data that can support compound profiling. Another aspect of neuroinflammation that's attracting quite a lot of interest at the moment is the inflammasome. So we have a system where we can look at inflammasome activation in the co-cultures as well. We use a fairly standard double protocol of LPS priming for 3 hours, followed by a 1-hour incubation with, in this case, nigericin. We've also looked at ATP. And then we quantify IL-1 beta release as well as IL-18. So we've demonstrated that in the co-cultures that see both priming and activation signals, we see a really robust increase in IL-1 beta release, and that you need both the priming and the activation signals to see this. The system is very reliable, very reproducible, to the point that we can generate dose response curves for both IL-1 beta and IL-18. I think, as you'd expect, most of the compounds that we've tested, the IC50s for IL-1 beta and IL-18 align pretty well. We also check that microglial nuclei counts remain constant, so these changes in cytokine release are not linked to the microglia being obliterated. And we're able to multiplex ELISA and imaging endpoints together to try and maximize data return. So before I wrap up, I just wanted to summarize really where stem cell models can support drug discovery. I mean, I'm slightly biased. I would say that they can support drug discovery at every stage. We certainly find that we're particularly supporting early target identification work. I think there's a huge power for novel target identification using these really disease-relevant systems. We've run target validation studies, but we have particularly, I think, added value at the late lead optimization and candidate selection phase, particularly with some of the compound profiling that I've described. Another point where I think we've run some really interesting projects is to look at life cycle management and opportunistic assessment of molecules that are maybe being developed for a different indication, maybe oncology or even respiratory, where we're interested in understanding whether or not they may have utility in neurodegeneration or neuroinflammation. And I think some of these systems have proved really useful just to generate data to either provide confidence that it's worth investing more resource, or to quash hopes at an early stage, or certainly to to provide data that would enable people to make a different decision about the way forward. So to summarise, I hope I've shown you that the human iPS-derived neuronal and glial systems are really valuable, but also very flexible models to support neurodegenerative disease research. In particular, the disease-relevant phenotypes can be quantified using a range of in vitro techniques. We particularly like the high-content imaging-based endpoints. And they're robust enough to support drug discovery. So it is possible to make very complex systems reliable and reproducible. The co-culture systems, so I've shown you microglia neuron co-culture models, but also tri-culture systems provide really interesting opportunities to explore cell-to-cell interactions in a robust in vitro system that is fully human. And these fully human systems are particularly well suited to validating gene therapies or any molecules where species cross-reactivity is not perfect, in addition to obviously providing that increased translational value. And I guess my final point is that these model systems are continually evolving. We're constantly trying to improve our understanding of the systems, as are many other groups. So it's an exciting space. And I just want to finish off by acknowledging all of the work of the Talisman science team, because I didn't generate these data, and they did, and they're really expert team and good at what they do. But thank you very much for listening.